Hey everyone! I want to start by thanking Erasmus Week organizers for creating this virtual platform and the opportunity to share our work. So today I'll be talking about the impacts of a changing ocean on sharks. And first of all, let me introduce myself. I am Dr. Maria Rita Pegado, and for the last 10 years I have been working at the Rui Rosa Lab at Laboratório Marítimo da Guia, which is this beautiful building with an amazing view of the ocean where we do most of our research. I have also added my Twitter account and my email address, so if you have any questions, feel free to reach out. So before I get into the shark science, I need to give you a little context about climate change and its impacts on marine life. If you have been watching the news, you can see many evidences of these climate changes occurring right now. From the snow in Brazil to the heat wave that hit Canada and the floods in Europe, China, and so on. And the main reason for these environmental changes is the increase of carbon dioxide in the atmosphere. Carbon dioxide occurs naturally in our planet, and it also is a really important greenhouse gas because it keeps the temperature of the Earth stable. So basically, as sunlight hits the planet, some of the energy goes back to space and some of it is absorbed and re-emitted as heat by greenhouse gases. This heat then is radiated in all directions to warm the planet. And so increasing the levels of carbon dioxide is like putting an extra blanket on the atmosphere, which is leading to a warmer planet. And thankfully, the oceans absorb most of the warming on land, but this warms up the oceans as well, causing ocean warming. And according to the IPCC, if we continue a business as usual path, the temperature of the oceans are expected to rise around 3 degrees by the end of the century. And we are already witnessing the expression of this warming through marine heat waves, which are already causing massive amounts of coral bleaching in many reefs around the world. And a marine heat wave, if you don't know, has been designated as a sudden warming event that occurs in a certain marine location when the upper local temperature threshold is exceeded for at least 5 days. And the frequency and duration of these marine heat waves has increased around the world, as you can see here, because each spike represents a marine heat wave. Here you can also see the longest and largest marine heat wave, which has lasted for 3 years and was designated as the blob. And this marine heat wave devastated phytoplankton growth. And as the impacts of marine heat waves can echo throughout the food webs, these episodes could disrupt entire marine food webs and reshape the biodiversity of the oceans. And ocean warming has an evil twin, unfortunately, because besides the capacity of the ocean to absorb the heat, it can also absorb the excess carbon dioxide that is present in the atmosphere, which contributes to decrease of the ocean's pH, causing ocean acidification. So basically what happens is that carbon dioxide enters the ocean and it reacts with seawater to form carbonic acid, which then quickly releases its protons to the seawater. As hydrogen protons increase, the pH decreases, which leads to ocean acidification. According to the IPCC, it is expected that the pH of the global ocean will further decrease around 0.3 units by the end of the century. And 0.3 units may not seem much, but it is actually a lot, because the pH scale is not linear, it is logarithmic. And this means that a small drop. So for example, from 8.2, which was the pH of the oceans before the Industrial Revolution, to 8.1, which is the current pH of the ocean now. This corresponds to a 26% of increase in acidity. And if we compare to 7.8, which is the pH expected by the end of the century, we are talking about a 151% increase in acidity. So the bottom line here is that small sounding changes in the ocean pH are actually quite large, and this may have severe consequences for marine life. And different organisms are affected in different ways by these environmental stressors. For example, calcifying organisms may be more impacted by ocean acidification since it reduces the amount of carbonate in the seawater, which is their key building block. On the other hand, if we're talking about tropical species, they may be more susceptible to ocean warming because they already live closer to their thermal limits. So understanding how these environmental changes may affect marine life can better help us comprehend a notion that it's already changing. So why is it important to study sharks? Well, first of all, the amount of research that has been focusing on the impacts of climate change in sharks is still scarce, as the literature is very asymmetric regarding the species that have been studied. Besides, having a K-Lab strategy compromises their ability to adapt to a rapid changing environment. And also, because their populations are severely decreasing, understanding if climate change also represents a threat is significantly important. And sharks have been roaming our oceans for a long time. And throughout this time, the concentration of carbon dioxide in the atmosphere has fluctuated a lot. So one could argue that climate change would only affect them indirectly. However, recent studies indicate that climate change may actually impact sharks both through direct effects on their biological responses, but also through indirect effects such as prey availability. So, as one of the most threatened vertebrate groups, understanding how they may respond to these climate changes represents a priority for their conservation. It is therefore important to understand how increased temperature and decreased pH may affect these species at different biological levels, so from molecules to cells and to the whole organism, which I will briefly explain in more detail. If we look into the molecular level, both warming and acidification can, for example, increase ROS production. And ROS are reactive oxygen species, which are these blue molecules that result from the partial reduction of oxygen. They are normally generated as byproducts of cellular metabolism, and while a small increase is important for several cellular functions, excessive ROS production can lead to oxidative stress. And this oxidative stress can cause oxidative damage in the proteins, the lipids, and also the DNA. So to determine these effects, we can measure the activity of antioxidant enzymes which are able to neutralize ROS, such as SOD, CAT, and also GPS. As an indirect measure of protein damage, we can also quantify HSP and ubiquitin, which stabilize and eliminate damaged proteins that result from this oxidative stress. Lastly, it is also possible to analyze the oxidative damage by measuring the lipid peroxidation and the DNA damage. And when we look at the cellular level, an increase of ROS production can lead to an increase in nuclear abnormalities of the blood cells, which include micronucleus, nuclear buds, and also binucleated cells, which impair the proper oxygen transport in the blood. It is also important to look at the hematological parameters in general, since warming may affect the membrane structure, the protein conformation, which may impair the proper oxygen transport, and the immune system. While the pH may affect enzymatic activity, which can also impair the oxygen transport and also the immune response. Lastly, at the organism level, all of these environmental stressors may affect growth, behavior, and even survival, among other things. So with that in mind, the objectives of our work were to analyze the impacts of a marine heat wave and ocean acidification on the hematological parameters and oxidative stress response of a temperate shark species. We also wanted to understand how ocean acidification could impact the growth rates, the swimming performance, the ventilation rates, and the acetylcholinesterase levels in the brain of a tropical shark species. So, starting with the marine heat waves, as I said, we wanted to observe its impacts on the hematological parameters and the oxidative stress response of a temperate shark species. To determine the parameters to study the marine heat wave, we needed to know the rate of onset, the maximum temperature registered for a marine heat wave, and the average duration. So for that, we used a data set of the local sea surface temperature for the last 30 years, and then calculated these values using an R package. We then obtained these values, which we used to plan and execute the experiment. So after an appropriate amount of time, we started to raise the temperature of the experimental group by 0.5 degree per day, which is the rate of onset. We did this for 6 days to reach a temperature of +3 degrees. And the experiment had lasted for 15 days, which was the average duration of a marine heat wave. So briefly, we exposed 12 small spotted cat sharks to control conditions at 18 degrees and another 12 sharks to a marine heat wave scenario at 21 degrees for 15 days. The first parameter analyzed was the ventilation rate, which was measured as the number of breaths per minute. And to get an average value, we measured the ventilation rate 3 times, and this was done before feeding to exclude any potential bias on the respiration. So the observations were then made at 3 different times. So before the experiment started, on the first day of warming, and after 15 days. As expected, exposure to a marine heat wave significantly increased the ventilation rates, and this happened immediately on the first day of the experiment and did not change during the 15 days. We then looked at the hematological parameters to understand the effects of a marine heat wave at a cellular level. So we counted the number of cells present in a blood smears, which included erythrocytes, both normal and with nuclear abnormalities. And the erythrocytes are the red blood cells, which are these ones. We also counted leukocytes, which are the white cells, so these ones. And thrombocytes, which are these cells, and they are similar to the human platelets. So for that, we used a drop of blood to perform the blood smears, which were then stained to allow better visualization of the cells. After that, a blind observer counted each cell using image-based software, and the number of blood cells were then compared between treatments. We also used the Newbar chamber to count erythrocyte cells. However, Because I was a PhD student with little experience on this subject, I wrongly interpreted and referred to the proportion of cells from the blood smears as cell counts. So basically, I disregarded the actual blood counts from the newborn chamber as redundant. And once I realized this mistake, I immediately analyzed the data, but the paper had already been published. Since it completely changed the results, I requested a correction and hopefully it will be republished soon. Unfortunately, these things happened, but I rather admit my mistake because I believe that it is crucial to improve scientific accuracy. Our results show the significant differences between treatments. So we saw a significant decrease of the proportion of erythrocytes, while we also saw a significant increase in the proportion of leukocytes and thrombocytes. However, as I said, these are only proportions. So if we look at the actual counts from the newborn chamber, we see a totally different trend. So we saw actually a significant increase of all the blood cells. So this means that increasing the temperature to simulate a marine heat wave also increased the demand for oxygen, either due to a higher metabolic need or a less oxygen available in the water, which resulted in an increase in erythrocytes without increasing the nuclear anomalies. We also saw the increase of leukocytes and thrombocytes, which may be acting as a primary defense in the immune response, since sharks' thrombocytes have also phagocytic capacities. We then analyzed several ratios, so the nucleus to cytoplasm ratio of the erythrocytes and the heart and spleen to body ratios. And we only found a significant decrease in the nucleus to cytoplasm ratio of the erythrocytes. So the ratio indicates the maturity of a red blood cell, because as these cells mature, the size of its nucleus decreases. So our results indicate that warming increases the amount of mature erythrocytes, which was also aligned with more erythrocytes that were counted, which means that there was more oxygen carrying capacity. Lastly, we took sample tissues from the gills, liver, and muscles to measure the oxidative stress response. And the samples were kept on ice and homogenized in PBS solution using an ultrathorax. These homogenates were then centrifuged, and the supernatant was then transferred to new Eppendorfs for further analysis. We then performed biochemical analysis in all of these samples to measure the antioxidant enzyme levels, which are the ones that neutralize the ROS formation, so the levels of SOD, CAT, and GPX. And although there were significant changes between tissues, we did not observe any significant differences between treatments. After, we measured the protein damage indirectly by analyzing the heat shock protein content and the ubiquitin. Again, there were no significant changes between treatments, although here I was expected that at least HSP would have been increased, since warming may alter the protein conformation. But sharks, they also are equipped with TMAO, which also functions as chaperones to refold damaged protein and may have enhanced here these sharks' thermal tolerance. I also have to add that these sharks perform vertical migrations in the water column as adults, and they face steep temperature changes. So these results suggest that even as juveniles, they can tolerate a 3-degree warming. Lastly, we measured lipid peroxidation and the DNA damage to determine oxidative damage caused by warming. Yet again, we did not observe any significant changes, which suggests that the species antioxidant system can cope with the warming trend of the present day, avoiding oxidative damage. Moving on to ocean acidification, we started by testing its impacts on the hematological parameters and the oxidative stress response of small spotted cat sharks. For that, we separated 18 sharks in control conditions where the pH was at 8.0 units and 18 sharks to treatment of acidification where the pH was at 7.7, and this was done for the whole embryogenesis plus 5 months. After that, the hematological parameters were assessed in the same manner as before. So briefly, we counted the erythrocytes, leukocytes, thrombocytes, and we also determined the ratios. Unlike what we observed in the marine heat wave, ocean acidification exposure did not change the proportion of any blood cells. It also didn't change the amount of erythrocytes with nuclear abnormalities. And lastly, the ratios did not change either, which suggests a certain resilience of these species towards ocean acidification. Next, we took some sample tissues from these sharks, including their gills, liver, and muscle. And then we again measured the oxidative stress response in the same manner as before. According to our results, there was only a significant increase in the CAT levels in the muscle tissue. This is possibly a result of increased ROS formation, such as hydrogen peroxide, because exposure to ocean acidification increases the amount of hydrogen protons, which can directly interact with water or the superoxide ion to form hydrogen peroxide, for which the CAT is the main antioxidant enzyme that neutralizes it. There were no significant changes in the HSP and ubiquitin levels. And even though there was an increase of CAT, there was no oxidative damage resulting from the exposure to a lower pH. And this suggests that these species may have a high tolerance to ocean acidification. In the last experiment, we studied a tropical shark species, the white-spotted bamboo shark. And tropical species are generally more vulnerable to warming because they already live closer to their thermal limit. For this reason, it is possible that these species migrate to colder temperatures. However, colder water tends to absorb more CO2, which can make them more acidic. With that in mind, we wanted to understand if ocean acidification could impact a tropical shark physiology. So for that, we used 10 white-spotted bamboo sharks, which we randomly assigned to either a control group or with a pH of 8.0 units and an acidification group with a pH of 7.7 units during 45 days after they hatched. We then assessed their Fulton condition and their specific growth rates, one considering the weight gain and another considering the growth. Our results showed a significant decrease of the growth rate based on length, which suggests that because it takes longer for them to grow, they may be more vulnerable for predation. We also observed their swimming performance using a swim tunnel, and we registered their maximum velocity, the time they spend swimming, and the number of bursts to increase the speed. The results showed a significant decrease in the time spent swimming. And as sharks rely on their swimming abilities to hunt and also to escape predators, this reduction may increase their vulnerability to predators and also reduce their hunting success, which in turn could further decrease their growth rates. We also measured their ventilation rates at 2 different times. So before and after a chase protocol, and before and after a swimming performance test. And the chase protocol basically consisted in the chasing of the shark by hand until it could no longer respond. So it was considered as exhausted. The results showed no significant changes between treatments. And the last analysis that we performed were to measure the acetylcholinesterase levels in 7 brain macro areas. This enzyme is responsible for breaking down acetylcholine, which is a neurotransmitter at the synaptic cleft. And this is important to effectively stop the neural signal. And this allows the compounds to be recycled and then rebuilt as a new neurotransmitter for the next message. The results showed a significant reduction of the activity in the optic lobes and in the telencephalon. So it is interesting to mention that there is evidence of visual input in a part of the telencephalon in sharks. The telencephalon is also considered an important coordinating center in elasmobranchs, responsible for influencing motor behavior and also multisensorial input, including vision. So with that in mind, the observed changes could not only be affecting sharks' motor behavior by reducing the swimming performance, but also their ability to properly respond to visual cues. Since we did not analyze this visual acuity, these results should foster more research. With this work, we showed that the temperate species was able to acclimate to ocean acidification with no evidences of any impact, while a marine heat wave elicited significant changes in the hematological parameters, but no evidences of oxidative damage. So here, warming appears to have a stronger effect on these animals' physiology than shifts in the pH. And since physiological compensation at the organism level is ultimately established through changes at the molecular level, the present findings suggest that the small spotted cat shark will be able to cope with sudden warming events that are occurring right now and expected high CO2 expected to occur in the future. On the other hand, ocean acidification has significantly impacted white-spotted bamboo sharks by reducing their growth, the swimming activity, and also the acetylcholinesterase levels on their brain. And this may be linked to energetic trade-offs, as the energy necessary to regulate the acid-base changes that are occurring may be prioritized in this case. So, even though these are sublethal effects, they still have the potential to reduce their individual performance with consequences over population dynamics. And this research is important to continue building our knowledge. And it is also essential to integrate these insights with similar research to fully understand the impacts of climate change in sharks. With that in mind, Catarina Santos and a team of researchers, including myself, we conducted an array of mixed model meta-analysis to evaluate these effects on elasmobranchs. In this meta-analysis, we compared climate change impacts over a set of biological responses, such as the ones that are listed in the table, to provide a quantitative overview of the existing literature and also to communicate the priorities for future research. I want to thank everyone at the Rui Rosa Lab who contributed for this research, and also, of course, to the funding agencies that made it possible. And I want to finish with my new project, which is entitled as Shark Rescue, which aims to protect sharks from overfishing by collecting their eggs. So if you are interested in knowing more about this, you are more than welcome to follow our progress. You can find us on Twitter or also on Instagram. And so that's it. I want to thank you all, and I am available for answering your questions. Thank you.